Hello students, welcome to this session. In this session we are going to discuss light reaction in photosynthesis under your chapter 13 NCERT biology 11th class. Light reaction. During last session we have studied that photosynthesis in higher plants is completed into two stages. First stage is the light reaction. Thereafter, the dark reaction occur in stoma, and light reaction occurs on thylakoid. Thylakoids are membranous sac-like structure on which the pigments are arranged in a specific pattern. During last session, somewhat we have discussed regarding arrangement of pigments in two types of photosystem. On thylakoids, the pigments are arranged in certain pattern. The arrangement of pigments on thylakoids is called as light harvesting complexes. On the basis of absorption capability of reaction center chlorophyll A, the light harvesting system may be classified into two types. Photosystem first and photosystem second. Photosystem first is also called as P700 and second is called as P680. In photosystem first, the reaction center, that is chlorophyll A, around the chlorophyll A, number of other pigments are arranged. Number of other pigments are arranged. All the pigments together assist the reaction center chlorophyll A in light reaction. So they are called as accessory pigments. Pigment like xanthophyll, carotenoids and other type of the chlorophylls also. They help the reaction center chlorophyll A in light reaction. So they are called as accessory pigments. Apart from the absorptions of different wavelengths of the light, these accessory pigments also save the chlorophyll A reaction center from the photooxidation. These accessory pigments absorb the lights of various wavelengths. So they provide a wider range of absorptions of the light. In photosystem first, The reaction center chlorophyll A absorb the red color of the light at maximum point. In red region, in this photosystem, the reaction center absorb the red color of light of having wavelength 700 nanometer. So it is also called as P700. While in photosystem second, the reaction center absorb the red color of the light of having 680 nanometer wavelength. So it is called as P680. These are the overall description of photosystem which take part in light reaction. Here, this light harvesting complexes. Thus, here we are finding the number of pigments are helping the reaction center chlorophyll A. And these accessory pigments are also called as antennae. Now we will discuss the light reaction. On the membrane of thylakoid, both type of photosystem are found. That means on the membrane of thylakoid, the absorption capability of the reaction center chlorophyll A is able to absorb the red light of 700 nanometer wavelengths as well as the red light of 680 nanometer wavelength. In context of participation of both the photosystem, we will understand the electron transport system. Electron transport system to generate the high energy chemical intermediate. High energy chemical intermediates are the main goal of the light reaction, because high energy chemical intermediate like ATP and NADPH plus H onward responsible for biosynthesis of carbohydrate in stroma of chloroplast. They are responsible for the reduction of carbon dioxide into carbohydrate. So synthesis of high energy chemical intermediates like ATP and NADPH2, or you may write like this also, are the main goal of light reaction. Now in context of participation of both the photosystem, we will discuss the electron transport system. In the case of participation of both the photosystem, the electron transport system is called as non-cyclic Photo-phosphorylation. In the case of respiration, certainly you have studied regarding phosphorylation. Phosphorylation is the process by which the chemical high energy intermediates are generated in the case of respiration. Similarly, in the case of light reaction of photosynthesis, these high energy chemical intermediates are generated in presence of light. So here the production of ATP and NADPH is called as photophosphorylation. Here the process is phosphorylations to generate ATP and NADPH. But because it takes place in presence of sunlight on the thylakoids of chloroplast, so it is called as photophosphorylation. In this case, the reaction center of photosystem second photosystem second firstly take part in trapping the solar energy or light energy. Here the reaction center chlorophyll A absorb the red color of light of having wavelength 680 nm. This is the P680. And after absorption of the light, solar energy, the electron get excited and jump, and here electron jump upward, uphill, and the excited electron is accepted by electron acceptor. Here is the electron acceptor, and thereafter the electron is transferred to the electron transport system. Electron transport system mainly consists of phytoquinine, cytochromes, phytoquinine and cytochromes. These are the electron acceptors which participate in electron transport system. Phytochrome, phytoquinine and cytochrome. At this stage, the photophosphorylation takes place and ATP is generated. ADP combines with inorganic phosphate and converts into ATP. This is high energy chemical intermediate. It is the currency of energy which is generated here at this stage. After that, the electron is transferred to the photosystem first. Photosystem first. It is also called as P700. As we know that in photosystem first, the reaction center chlorophyll A absorb the red color of the light having 700 nanometer wavelength at maximum point. And after absorption of the red light at maximum point, the electrons of reaction center get excited. Here the electron received from the electron transport system also increase the excitement of this reaction center, and by this way here the redox potential is generated at great extent. And after that, the excited electron jumps upward, uphill. Here the electron after this point move downward, downhill movement up to here. And after the photosystem first, the electron having great redox potential jump upward, and again here it is received by electron acceptor. And thereafter, after receiving the electrons of great redox potential, the electrons again move downhill. And here reduction of NADP takes place, and by this way this reducing agent is produced. It is also maybe written like this, but generally we write by this way. This is the right way to represent the NADPH2. And at the end, in this non-cyclic photophosphorylation, ATP here and one NADPH2 is generated, and it is a continuous process. Here the replacement of, because the electrons after getting excited jump upward, so its loss is compensated by splitting of water. During splitting of water in presence of light, electrons are produced. This splitting of water in presence of light is called as photolysis. Here, by the process of photolysis, the loss of electron is compensated by photolysis, splitting of the water. And this is the continuous process. So, this is the process by which these reducing agents ATP and NADPH are produced continuously. And this combination of the two high energy chemical intermediate are together called as assimilatory power. Assimilatory power or reducing agent, because after light reaction these assimilatory power ATP and NADPH enter into a stroma of chloroplast, where they reduce the carbon dioxide into C6H12O6. Biosynthesis of glucose or carbohydrates takes place in the stroma, and in reduction of carbon dioxide, they are the main factors. So they are called as assimilatory power or reducing power. This is the overall description of non-cyclic photophosphorylation. And non-cyclic photophosphorylation takes place on membrane of thylakoid. As the membrane of the thylakoid consists of both the photosystem first and second, but on stroma lamellae, you know, these are the thylakoids and this group is grana, and thylakoids are interconnected by membranous tubular structures in chloroplast, and this is called as the stroma lamellae. On stroma lamellae, only one type of the photosystem is found, that is the photosystem first. That means on stroma lamellae, the reaction center of the photosystem chlorophyll A only absorb the red light of having 700 nanometer wavelength at maximum point. Maximum absorption by the reaction center of the photosystem is the red light of 700 nanometer. So, at stroma lamellae, the cyclic photophosphorylation takes place because only one photosystem participates in light reaction. We will discuss here. The cyclic photophosphorylation. By this way, here we are finding that in light reaction, firstly the light energy is trapped in chloroplast. Thereafter, water splits to generate electrons. Oxygen is generated and reducing agents or assimilatory power are generated to participate in dark reaction onward. Now we will discuss the cyclic photophosphorylation. It takes place at stroma lamellae. We know the stroma lamellae. In this case, the photosystem first only participate. Only photosystem first, that means P700 is found on stroma lamellae. After absorbing the red light at maximum wavelength of 700, the electrons of reaction center get excited, and after excitement it jump upward and the electrons is received here by electron acceptor. Clear? Upward movement. And thereafter the electron from the electron acceptors is received or moved downward, downhill, and received by the electron transport system consisting of cytochrome, phytoquinone, or cytochrome. And here, just similar to the non-cyclic photophosphorylation, here ATP is generated from ADP and inorganic phosphate, and thereafter the electron returned back to the photosystem first again. Thus here the movement of electron is cyclic in the case of photosystem first, and because here only photosystem first is participating. So in the case of cyclic photophosphorylation, the electron move in cyclic way, again return back to the points from where it starts the movement. Clear? This is cyclic. And by this way we completed both non-cyclic photophosphorylation and cyclic photophosphorylation. In next session we will discuss the push-up of assimilatory power into the stroma. And to understand the push-up of assimilatory power from the stroma lamellae as well as from the thylakoids into the stroma, we will study the chemiosmosis hypothesis, by which we will understand how the assimilatory power ATP and NADPH are pushed up into the stroma to participate in the dark reaction. By this lecture I am concluding. You students continue with us. Watch the videos available on our YouTube channel. You subscribe the Institute channel, get benefited as well as help the Institute in promotion of Institute's YouTube channel. Thank you very much.